Hello children. Today I am going to start with a topic that is how Mendel proved the laws of inheritance. As already discussed in the previous topic, we have learned about various terminologies which Mendel used while performing his experiments. Now today I am going to detail you about the cross which Mendel performed to prove the various laws of inheritance. Now first of all, we will know how Mendel started with his experiment. Mendel did his work on various plants, but finally he chose the pea plant, his garden pea plant. What was the reason? Why did he choose the pea plant? The scientific name for the pea plant is Pisum sativum. So why did he choose the pea plant? There were various reasons that why he chose the pea plant. Number one, these plants were easily available. They were able to produce bisexual flowers. Number third, they were able to self-pollinate, and even cross-pollination was also possible. These plants, they produced large number of seeds from each plant. Then another reason was these plants were annual plants, means he was able to see several generations within his tenure when he started working on these laws of inheritance. Now, finally, how he came to the conclusion, he selected 7 contrasting characters from these pea plants. He did hundreds of crosses, and finally he got the pea plant with 7 contrasting characters. What were those 7 contrasting characters? He took the consideration the seed color. What was the contrasting character which he was able to find? It was yellow and green. The color of the seed was yellow and green. Seed shape, second contrasting character, he was able to find round and wrinkled seeds. Pea plants which were having the shape that is round and wrinkled. Seeds which were having the shape round and wrinkled. Then flower color, it was violet and white. Pod color, yellow and green. Pod shape, inflated and constricted. Flower position, axial and terminal. And stem height, tall and dwarf. These were the 7 contrasting characters which Mendel found while doing his crosses. He worked on the law of inheritance, and these laws there were 3 laws, that is law of dominance, law of segregation, and law of independent assortment. Now, how he proved these laws? He focused in his first cross, where he focused only on one character. He took into consideration only one character. And how he performed his experiment, he called it as monohybrid cross. Monohybrid cross. What do you mean by mono and hybrid? Mono means one, and what do you mean by hybrid? A hybrid is a new form of plant resulting from a cross or breeding between 2 plants to get a new combination. So hybrid is a new form of plant which results crossing between 2 plants of the same species. So he took one character into consideration and he focused on one character. What was that character? The character was height. He took one character that was height in his monohybrid cross. Now, he focused on the character height. he took the pure variety. As you know that pea plants, they are bisexual flowers, they show self-pollination. So he was not able to get the pure variety when these plants self-pollinated. So what he did emasculation. What is emasculation? Emasculation means removal of anther from the flower before maturity. So he removed, to get a pure variety of the plants, pea plants, he removed the anther from the flower before maturity, and when the flower became mature, he dusted the pollens of the required variety. He dusted the pollen from other pure variety to get a pure hybrid. So what he did, he took the parental generation pure tall and pure dwarf. Pure tall and pure dwarf. And he represented these pure tall and pure dwarf with the letters or the symbol. He represented the pure tall with the capital letter and pure dwarf with the small letter, small t. Now, what were the gametes? As you know, gametes have half the number of chromosomes. So what will be the gametes? These were the gametes which were produced. Now, F1 generation, as already explained you, what is F1 generation? The first filial generation, or the F1 progeny, or the young ones which are produced. So, in the F1 generation, he got the plants which were all tall. Which were all tall. This was the phenotype. Phenotype means the physical appearance. But what was the genotype? Genotype, none of them were pure tall. All of them were in a heterozygous condition. That means they had the genes both for the tallness and the dwarfness. But if you will see the parental generation, there were the parents were pure tall and pure dwarf. But if you see the F1 generation, all of them were tall, physically tall, but none of them was pure homozygous, in a homozygous condition. All of them were heterozygous. So the genotype was, they all were heterozygous. So what does this indicate? You have seen the parental generation were pure tall and pure dwarf, but the F1 generation, although physically all of them were tall, but none of them was genotypically none of them was pure tall. So what does this indicate? This indicates the first law of dominance. What is the first law of dominance? The first law of dominance is the gene which expresses itself in a heterozygous condition, or the gene which expresses in presence of an alternate gene for the same character is called as the dominant gene, and the one which does not express itself in presence of an alternate gene for the same character is called as the recessive. So the first law of inheritance, that is law of dominance. What is law of dominance? The gene which expresses, the character which expresses itself in presence of an alternate gene for the same character is called as the dominant gene. In presence of an alternate gene for one character, in presence of an alternate gene for one character is called the dominant gene. This was the first law of inheritance given by Mendel. So, we came to know what is a law of dominance. Now, then he allowed the F1 generation to self-pollinate. He allowed the F1 generation to self-pollinate. What was F1 generation? F1 generation, the plants were tall but in a heterozygous condition, So, F1 generation, capital T, small t, capital T, small t. Now, gametes, what will be the gametes? Half the number of chromosomes, that is capital T, small t, capital T, small t. Now, the F2 generation. What was F2 generation? Capital T, capital T, capital T, small t, capital T, small t, small t, small t. So, if you will see, in F2 generation, the pea plants which were produced, they were in a different combination. Number one, tall, pure tall. This was again tall. The phenotype. What was the phenotype? 3 were tall and 1 was dwarf. So, what was the phenotypic ratio? 3:1. 3 plants, pea plants were tall and 1 pea plant was dwarf. This was the physical appearance, the phenotypic ratio. But when he found the genotype, what was the genotypic ratio? 1 was pure tall, 2 were in a heterozygous condition, and 1 was pure dwarf. This is the genotypic ratio of a monohybrid cross. The genotypic ratio of a monohybrid cross is 1:2:1. 1 pure tall, 1 heterozygous, 2 heterozygous tall, and 1 pure dwarf. Now, if you will see the F1 generation and the F2 generation and the parental generation, In the parental generation, you were having pure tall and pure dwarf, but you were not able to find that pure tall and pure dwarf in F1 generation. But the genes for both tallness and dwarfness were present there, but they were not able to express. The gene for dwarfness was not able to express because it was a recessive gene. But in F2 generation, the gene which was for pure tall was able to express itself in the F2 generation. This means that the genes, parental genes or the parental characters are present in their successive generation, but sometimes they remain hidden. They cannot express themselves, but they will be expressed in next generation, which we can see in F2 generation. So, what was the second law of inheritance? The second law of inheritance was law of segregation or purity of gametes. What was the second law of inheritance? Law of segregation or purity of gametes. What is law of segregation or purity of gametes? Means when a pair of factors are brought together, when a pair of factors are brought together at the time of gamete formation, at the time of fertilization, they do not mix or blend together. They remain in the pure form. They do not form a new character. What is law of segregation of purity or purity of gametes? When the factors for a particular character at the time of gamete formation or at the time of fertilization are brought together, they do not mix or blend together, but they remain as they are, which you can see this parental generation was absent in F1 generation, but you can find this parental generation in F2 generation. This means the character for tallness and the character for dwarfness, they have not mixed together, although they were there, but they have not created any other character. They are there as they are, but the one which expressed itself is the dominant one, and the one which was not able to express itself is the recessive one. So, what is law of independent assortment? When a pair of contrasting factors or genes are brought together in a hybrid, these factors do not blend or mix together. These factors do not blend or mix together. They remain as they are. This is the second law, that is law of segregation or purity of gametes. So, the two laws, law of dominance and law of inheritance, law of dominance and law of segregation, were proved by Mendel in his first cross, that is the monohybrid cross, where he focused on one character. And what was the character? That was the height of a plant. I repeat it once more. The law of segregation, it proves that the factors or the genes for a pair of they remain, they assort, they segregate independently, and they do not mix or blend together and separate independently at the time of gamete formation. Now, the third law of inheritance, what was that? The law of independent assortment. The law of independent assortment. For this, Mendel did a dihybrid cross. Mendel performed a dihybrid cross. What do you mean by di? Di means two. So, in this cross, he focused on two characters. In monohybrid cross, he focused on only one character. What was the character? That was height. But in his dihybrid cross, at the same time, he focused on two characters of the pea plant. What were those two characters? the shape of the seed and color of the seed. Again, he took the pure variety of the pea plant, the parental generation. The parental generation. He took the pure variety, that is the seed, the plant with round and yellow seeds. Round shape and yellow seeds. Yellow color. Seeds with round shape and yellow color. And he took the another parent with in parental generation, he took the pure variety of the pea plant with round seeds with round shape and yellow color, and he represented it with the symbol, that is capital R, capital R, capital Y, capital Y. Capital R for the round shape and capital Y for the yellow color. And another parent, he took the pea plant with wrinkled shape and green color seeds. That is, he represented it with the small letter, that is small r, small r, small y, small y. These were the pure varieties. So at a time he focused on two characters. What were the two characters? The shape of the seed and the color of the seed. So the gametes were round yellow and wrinkled green. These were the gametes. So what was the F1 generation? So in F1 generation, he found that all the plants, pea plants, in F1 generation he found that all the pea plants were round and yellow. Round and yellow. F1 generation, all the pea plants were round and yellow. But again in a heterozygous condition. None of them was the pea plant with round and yellow seeds. All of them were in a heterozygous condition. Again this proved the law of dominance. Then he allowed the plants of F1 generation to self-pollinate. He allowed the plants of F1 generation to self-pollinate. F1 generation, all the pea plants were round and yellow. Were having the seeds which were round and yellow. So they were Mendel allowed them to self-pollinate. So the gametes were round yellow, round green, round wrinkled yellow and wrinkled green. So the statistical ratio was not able to be done with a simple cross. So there was a scientist who made a square that is called as the Punnett square, where we were able to find the statistical ratio. What was the ratio? What were the different varieties which were produced in this dihybrid cross. There was a scientist, his name was Punnett, who made the Punnett square to show the statistical ratio of the F2 generation in a dihybrid cross. So, what was this, how he made it? So, what were the gametes? Round yellow, round green, wrinkled yellow and wrinkled green. This was the male gamete and the female gamete. Round yellow, round green, wrinkled yellow and the wrinkled green. So, the gametes, when they fused together, this was the product which Mendel got after allowing them to self-pollinate self-pollinating the F1 generation. So this was the F2 generation. So what was the number? In F2 generation, there were the pea plants with round yellow seeds. There were the pea plants with round green seeds. There were the pea plants were produced which were having wrinkled yellow seeds, and the pea plants with wrinkled green seeds. How many round yellow seeds were produced? It was 9. The ratio was 9:3:3:1. Now, if you can see, in the parental generation, there were only 2 combinations. The pea plants with pure round and yellow seed, and the pea plants with pure wrinkled and green seed. And in F1 generation, we got the pea plants which were round and which were having the seeds with round and yellow color. But these were not in a homozygous condition, they were in a heterozygous condition, but their parents were pure. And then when Mendel allowed them to self-pollinate the F2 generation, in F2 generation we could find out new combinations. What were the new combinations? That the seed which was having round shape, it has the green color, and the seed which was having wrinkled shape, it has the yellow color, which was nowhere in their parental generation or F1 generation. So what does this prove? This proves the law of independent assortment. So what is the law of independent assortment? The law of independent assortment is the genes for different characters, they assort independently. They are not dependent on each other. They assort independently at the time of their gamete formation. What do you mean by that? If you will see that in the parental generation, we were having a pea plant which was having round and yellow shape, and we were having a pea plant which was having wrinkled and green color. But in F1 generation, again we had the same combination, but not in a pure variety, but it was in a heterozygous condition. But in F2 generation, we got the new combination. This means that the genes for a particular character, although they are together in one generation, but they assort independently at the time of gamete formation. They are not, these characters are not dependent on each other. This is what is called as the third law, that is law of independent assortment. Now we have read about the monohybrid cross and the dihybrid cross, where the three laws have been, law of inheritance has been proved. What was the first law? That is the law of dominance. You have seen what is a law of dominance? When a gene expresses itself in presence of an alternate gene for the same character, it is called as the dominant gene, which you have seen in the monohybrid cross. And what was the ratio in a monohybrid cross? The phenotypic ratio in a monohybrid cross was 3:1, and the genotypic ratio was 1:2:1. This was monohybrid cross, and in monohybrid cross again the second law was proved, that is law of segregation. What is the law of segregation? When a pair of contrasting factors or genes are brought together in a hybrid, these factors do not mix or blend together. They simply associate themselves with each other and separate at the time of gamete formation. And the law of independent, the third law, which was proved by Mendel in his dihybrid cross, where he took two characters into consideration. What happened in, what was the ratio? What was the ratio of a dihybrid cross? It is 9:3:3:1, where we have observed that two different combinations arised in the F2 generation, which were not present neither in the parental generation nor in the F1 generation. So the third law, that is law of independent assortment, the genes of different characters. So two characters were considered. The genes of different characters located in different pair of chromosomes are independent of each other. They do not depend on each other. This means the yellow color was not dependent on the round shape, and the wrinkled shape was not dependent on the green color, because we got the combinations in F2 generation which were yellow and wrinkled and green and round. So this is what is law of independent assortment. That means the genes of different characters located in different pair of chromosomes are interdependent on each other and assort independently at the time of gamete formation. These were the laws of inheritance which were given by Mendel. Now coming to sex determination. So the next topic is sex determination. If we talk about humans, in humans there are two types of chromosomes. One type of chromosomes, they are called as the autosomes. And the another type, that is called as the allosome. What are autosomes? Autosomes are the chromosomes which determine the normal functions of human body, except for sex determination. And what are allosomes? Allosomes are also called as the sex chromosome, which determine the sex of an individual. How many number of chromosomes are there in a normal number of chromosomes are there in a human body? There are 23 pair of chromosomes in humans, out of which 22 pair are autosome and the 23rd pair is the allosome, the sex chromosome. So there are 23 pair of chromosomes in human body. Now how can we determine the sex of an individual, whether it is a male or a female? So how can we determine the sex of an individual, whether it is a male or a female? The males, they have 22 pair autosome plus the 23rd pair is XY, which is called as the sex chromosome. Females, they have again 22 pair of autosome plus the XX, which is called as the sex chromosome in females. Now, how can we identify that whether a young one is a male or a female? Males 44 plus, if it is a male, 44 plus XY, and females female 44 plus XX. What will be the gametes? 22 plus X and 22 plus Y. What will be the gametes here in female? 22 plus X and 22 plus X. So, the young one can be 44 plus XX, 44 plus XX, 44 plus XY, and 44 plus XY. So, what is the probability? The probability is that 50% of the progeny can be, there will be 50-50 chances of having either a boy child or a girl child. If you see, you have 50:50 ratio. So this probability depends upon the father, whether he has the X chromosome in the sperm or the Y chromosome in the sperm, which can fuse with the mother's ova, and we can have the chances of either a boy or a girl. So usually in the family, it all depends upon the probability. Basically, genetics is all the chance, the probability which we can see in the young ones. So, being a chance that a young one can be a girl or a boy, the ratio in a family will be different. It all depends upon the father, whether it has the sperm with the X chromosome or the sperm with the Y chromosome. Because the females, they have the sex chromosome, both the sex chromosomes are, the genes for the sex chromosomes are same. So with this we have concluded, we have completed the topic, that is heredity. Now in our next class we will be starting with the topic that is evolution. Thank you.